Development and aging. In the course of its life, an organism changes from a fertilized egg into an adult. This process of conversion from simpler to more complex form is called development. As development proceeds, all sorts of changes take place. The most obvious change is growth. However, these positive changes become negative at some stage in the life cycle, which are termed as aging. Embryonic development. The progressive changes which are undergone before an organism acquires its adult-like form constitute the embryonic development. It begins with a series of mitotic division in the zygote. These early divisions of the zygote are called cleavages. Early cleavages and blastocyst formation. Cleavage is a period of fairly rapid mitotic division of the zygote following fertilization. Some 36 hours after fertilization, the first cleavage division has produced two identical cells called blastomeres. These divide to produce four cells, then eight, and so on. As a result, a loose collection of cells that form a berry-shaped cluster of 16 or more cells called the morula has been formed. Zygote, two cells, four cells, multicells, morula, blastocyst. All the while, transport of the embryo toward uterus continues. By day 3 or 4 after fertilization, the embryo consists of about 100 cells and floats free in the uterus. By this time, it has tightened its connection between neighboring cells, process called compaction, and begins accumulating fluid within an internal cavity, the zona pellucida. The external membrane now starts to break down, and the inner structure, now called blastocyst, hatches from it. The blastocyst is a fluid-filled hollow sphere composed of a single layer of large latent cells called trophoblast. Cells and small cluster of 20 to 30 rounded cells called inner cell mass located at one side. Trophoblast cells begin to display L-selection adhesion molecules on their surface sown. After the blastocyst hatching, they also take part in placenta formation and secrete and display several factors with immunosuppressive effects that protect the trophoblast, hence the developing embryo, from attack by mother cells. The inner cell mass becomes the embryonic disc, which forms the embryo and the extra embryonic membrane except the chorion and trophoblast derivative. Implantation of early embryo. Implantation of early embryo, the extra embryonic membrane that form during the first 2 to 3 weeks of development include the amnion, yolk sac, allantois, and chorion. The amnion develops when cells of the epiblast fashion themselves into transparent membranous sac. This sac, the amnion, becomes filled with amniotic fluids. Later, later as the embryonic disc curves to form a tubular body, the amnion curves with it. Eventually, the sac extends all the way around the embryo, broken only by umbilical cord. Gastrulation. During week 3, the two-layered embryonic disc transforms into three-layered embryo, in which the primary germ layers ectoderm, mesoderm, and endoderm present. This process called gastrulation involves cellular rearrangements and migrations. Gastrulation begins when a groove with raised edges, called primitive streak, appears on the dorsal surface of the embryonic disc and establishes the longitudinal axis of the embryo surface. Epiblast cells of the embryonic disc then migrate medially across other cells and enter the primitive streak. The first cells to enter the groove displace the hypoblast cells of the yolk sac and form the most inferior germ layer, the endoderm. Those that follow push laterally between the cells at the upper and lower surfaces, forming the mesoderm. The cells that remain on the embryo's dorsal surface are the ectoderm. The mesodermal cells immediately beneath the early primitive streak aggregate, forming a rod of mesodermal cells called the notochord. The first axial support of the embryo. At this point, the embryo is about 2 millimeters long. The three primary germ layers serve as primitive tissues from which all body organs will derive. Ectoderm fashions structures of the nervous system and the skin epidermis. Endoderm forms the epithelial linings of the digestive, respiratory, and urinogenital system and associative glands. Mesoderm forms virtually everything else. Gastrulation lays down the basic structural framework of the embryo and sets the stage for the rearrangements of organogenesis, formation of the body organs and organ systems. Neurulation, neurula formation. The formation of organs and systems during embryonic development is called organogenesis. The first major event in organogenesis is neurulation, the differentiation of ectoderm that produces the brain and spinal cord. This process is induced by chemical signals from the notochord, the rod of mesoderm that defines the body axis. The ectoderm overlying the notochord thickens, forming the neural flat, and then starts to fold inward as neural groove, which forms prominent neural fold as it deepens by day 22. The superior margins of the neural fold fuse, forming a neural tube. It soon pinches up and becomes covered by surface ectoderm. The anterior end of the neural tube becomes the brain and the rest becomes the spinal cord. The associative neural crest cells migrate widely and give rise to the cranial, spinal, and sympathetic ganglia and associated nerves, to the medulla of the adrenal gland, and to segment cells and contribute to some connective tissues. By the end of first month development, the three primary brain vesicles, fore, mid, and hindbrain, are obvious. By the end of second month, all brain fractures are evident. The cerebral hemispheres cover the top of the brain. Stem and brain waves can be recorded. Most of the remaining ectoderm forming the surface layer of the embryonic body differentiates into epidermis of the skin. Control of development. We know that every gene is present in every cell of developing embryo. Mitosis guarantees that every daughter cell of the zygote will have full set of chromosomes with all of their genes. As the development proceeds further, we find that different cells differentiate along different lines and perform different functions. For example, one enzyme produced by the cell of stomach is useful in digestion of food. Another enzyme produced in cell of fingers and toes forms the protein keratin of the fingernails and toenails. We are quite sure that genes governing the production of both these enzymes are present in the cell of stomach and those of the fingers and toes. Why one kind of cell forms one sort of enzyme while the other kind of cell forms a different enzyme? The most logical answer to this question is that if the nuclei are identical in these cells, then perhaps the cytoplasm may vary. Thus, both the nucleus and cytoplasm play an important role in normal development. The nucleus determines the characteristics of the individual, while the cytoplasm selectively turns on some genes and switches off others. Rule of nucleus and cytoplasm and development is shown with the help of the following simple experiments. Rule of nucleus and development. Importance of nucleus and development was expressed in unicellular alga. The Acetabularia, it has 2 to 3 inches in length, inhabits European seawater. It has stalk with a characteristic cap and base containing nucleus. The two species of this alga are different in shape of their Stabularia mediterranea has umbrella-shaped cap while Stabularia crenulata has irregular head. The cap of both species were cut and thrown away. Their stalk were also removed but exchanged. The stalk of Stabularia crenulata was grafted on the base of Stabularia mediterranea. And vice versa. A whole new alga forms from the jointed pieces. It was found that new regenerated according to the type of nucleus present in the base. The nucleus exerts a strong influence on the development of up through the messenger RNA. This is evidence for nuclear control and developmental process. Role of cytoplasm. The gray crescent in cytoplasm, director of cell destiny. This experiment demonstrates that the cytoplasm and the crescent-shaped gray area of fertilized egg frog zygote helps direct embryonic development. Normal cleavage divides the zygote through the gray crescent, so each daughter cell receives an equal share of the components in the cytoplasm. If these two cells are experimentally separated, each grows into normal tadpole. However, if the first division is altered so it doesn't bisect the gray crescent equally, only the cell containing the gray crescent develops normally and the one lacking gray crescent develops into an undifferentiated ball of cells. It is known that different cytoplasmic components contain different morphogenetic determinants that are responsible for cell differentiation. These determinants are present in blastomeres. For example, the fertilized egg of an ascidian contains cytoplasm of 4 different colors that are segregated into different blastomeres. Clear cytoplasm produces larval epidermis. Yellow cytoplasm gives rise to muscle cells. Gray vegetal cytoplasm gives rise to gut. Gray equatorial cytoplasm produces notochord and neural tube. Mechanism of cellular determination. How do cells become different from their parent cells? How do two identical daughter cells become different from one another? How might one daughter cell become a neuron while the other daughter cell becomes a skin cell? In some cases, determination results from asymmetric segregation of cellular determinants. However, in most cases, determination is a result of inductive signaling between cells. Asymmetric segregation of cellular determinant is based on asymmetric localization of cytoplasmic molecules, usually proteins or messenger RNAs, within a cell. Before it divides during cell division, one daughter cell receives most of all the localized molecules, while the other daughter cell receives less or none of these molecules. This results in two different daughter cells, which then take on different cell fates based on differences in gene expression. Induction. Although there are many examples where asymmetric segregation of cellular determinants leads to differences between daughter cells, more frequently we find that cells become different from one another as a result of inductive signals coming either from other cells or from their external environment. There are many examples in development where an inductive signal from one group of cells influences the development of another group of cells. There are three main ways in which signals can be passed between cells. In first mechanism, a diffusible signal is sent through the extracellular space and is received by a cell surface receptor, which further transmits the signals by way of second messengers. In the second mechanism, cells directly contact each other through transmembrane proteins located on their surfaces. In the third mechanism, the cytoplasm of two cells is connected through gap junctions, allowing the signal to pass directly from one cell to another cell. And plant direct connections between cells are called plasmodesmata. In the 20th century, the German zoologist Hans Smann and Hald Mangold discovered in an early gastrula, an extremely important morphogenetic field with amazing properties. Their experiments involved dissecting a small piece of tissue from the dorsal lip of the blastopore in an early gastrula of a newt and transplanting it to the opposite side of another gastrula. The result was formation of second notochord and neural tube during gastrulation and neurulation in the host embryo, and ultimately the formation of an entirely new body axis. The resulting embryo developed two bodies, though technically difficult because the gastrulas were only about 2 mm wide. These experiments yielded clear results because the researchers used two very closely related species of newts. Triton tenatus, which is pigmented, serves as tissue donor, and Triton cristatus, which is non-pigmented, served as the host embryo by transplanting pigmented tissue from the donor embryo into non-pigmented host embryo. Spearman and Mangold were able to visually track the origin of the newly developed tissue. The results showed that the secondary notochord and neural tube were composed in large part of host non-pigmented cells, with some pigmented cells remaining from the transplanted tissue. This indicated that the transplanted tissue, which they named the organizer, had induced cells in the host to differentiate into neural tissue. On the transplanted side of the embryo, more recent work has shown that organizer secretes morphogens. Human embryonic development. A normal pregnancy lasts 9 months. Each 3 months period of pregnancy is called trimester. During each trimester, the fetus grows and develops. Fetal development, the first trimester, week 1 to 12. Soon after fertilization, the zygote travels down to the fallopian tube toward the uterus. At the same time, it will begin dividing rapidly to form cluster of cells resembling a tiny raspberry. The inner group of cells will become the embryo. The outer group of cells will become the membrane that nourish and protect it. Implantation takes place in the fourth week of pregnancy. By the time it reaches the uterus, the rapidly dividing ball of cells, now known as blastocyst, has separated into two sections. The inner group of cells will become the embryo, the outer group of cell will become the membrane that nourish and protect it. On contact, it will burrow into the uterine wall for nourishment. This process is called implantation. The placenta, which will nourish the fetus throughout the pregnancy, also begins to form. The fifth week of pregnancy marks the beginning of the embryonic period. This is when the baby brain, spinal cord, heart, and other organs begin to form. The embryo is now made of three layers. The top layer, ectoderm, will give rise to the baby outermost layer of skin, central and peripheral nervous system, eyes, inner ear, and many connective tissues. Now, baby's heart and primitive circulatory system will form in the middle layer of cells. The mesoderm, this layer of cell, will also serve as the foundation for the baby's bones, muscles, kidneys, and much of the reproductive system. Growth is rapid. In coming weeks, the neural tube along fetal back is closing and heart is pumping blood. Basic facial features will begin to appear, including passageways that will make up the inner ear and arches that will contribute to the jaw. The fetal body begins to take on C-shaped curvature. Small birds will soon become arms and legs. Tiny nostrils become visible and the eyes lenses begin to form. The arm buds now take on the shape of fiddles. The arm and legs are growing longer and fingers have begun to form. The external part of the baby ears also are forming and eyes are visible. The upper lip and nose have been formed. The trunk of the body is beginning to straighten. To the end of first trimester, the embryo can be now technically described as fetus. Fetus is developing flesh, which is the most fat gain throughout the pregnancy. The fetus will be growing the most rapidly during this stage, gaining up to 28 grams per day. The fetus begins to move regularly and expelled by a woman. Movement of the fetus becomes stronger and more frequent, and with improved brain, eye, and muscle function, the fetus is prepared for ex utero viability. This is head engagement in the third trimester. That is, the fetal head descend into the pelvic cavity. 40 weeks into pregnancy or 38 weeks after conception, baby might be about 18 to 20 inches long and weigh 6 to 9 pounds. However, healthy babies born in different sizes are just as normal to deliver a baby a week or too late or early as it is to deliver on due date. Twins and quadruplets. A pregnancy of two or more fetuses is called multiple fetuses. Multiple fetuses can be the same, identical, or different, fraternal. Identical twins or triplets come from a single egg that has been fertilized by one sperm. For unknown reason, the fertilized egg splits into two or more embryos during the first stage of development. Fraternal multiples come from multiple fertilized eggs. Some identical multiples share the same placenta, however, they usually grow within separate amniotic sacs in the uterus. In rare cases, identical multiples share one amniotic sac. Fraternal fetuses have separate placentas and amniotic sacs. Placentation. Some cells of embryo grows into a disc-like structure called placenta. The placenta closely attached to the embryo by a tube called umbilical cord. The umbilical cord connects the embryo to the placenta. The blood vessels of the placenta are close to those of uterus so that oxygen, glucose, amino acid, and salt can pass from the mother's blood to the embryo's blood. In a similar way, carbon dioxide and urea in embryo's blood escape in the placenta and are carried away by mother blood in uterus. The placenta can prevent some harmful substances in the mother blood from reaching the embryo. It also produces hormones like estrogen and progesterone, which helps in maintaining the pregnancy and prepare the mother for delivery. It also helps in the periodic contraction of uterus during delivery. A few weeks before the birth, the head of the fetus is turned down in the uterus, just above the cervix. Childbirth begins when the muscular layers of uterus start to contract and relax. These actions are felt as labor pains. Opening of the cervix widens enough to let the baby head pass through. Head of the baby acts as wedge to assess cervical dilation. The amniotic sac breaks at some stage in labor and the fluid escapes. Finally, the baby is pushed through the vagina, now called birth canal, and to the outer world. The birth occurs. The umbilical cord is cut and tied. After the birth, a baby contraction of uterus continues, during which the placenta is expelled. The sudden fall in temperature felt by a newly born baby stimulates it to take its first breath and thus usually cries. In a few days, the remains of the umbilical cord attached to the baby's abdomen shrivel and fall away, leaving a scar in the abdominal wall called the navel. Lactation. The newborn baby is supplied with maternal milk soon after its birth. The mammary glands of mother are especially prepared during the period of pregnancy. Under well-defined hormonal control, secretory ducts fed in the mammary gland branch further and undergo enormous development and start producing milk by the end of the pregnancy. The milk produced initially by mammary gland contains special lymph-like fluid known as colostrum, which is quite rich in antibodies. Usually, the baby is fed on the maternal milk for up to 2 years. However, depending upon the general health condition of the mother as well as other physiological conditions, maternal milk may not be available for so long. The baby is then fed on other sources of milk. As soon as the mother stops feeding the baby, her reproductive cycle begins again. However, sometimes the reproductive cycle can initiate even when mother is breastfeeding. Breastfeeding versus bottle feeding. There are advantages and disadvantages to both breastfeeding and bottle feeding. Advantages of breastfeeding. Breast milk has perfect balance of nutrients. It is easily digested and absorbed. Breast milk is always at perfect temperature. Milk is readily available at any time and any place. Advantages of bottle feeding. The only advantage is anyone can feed the baby. Presence of mother is not necessary. Disadvantages of bottle feeding. It can create unhygienic circumstances for the baby. It is vulnerable to carry various types of infections. It can cause indigestion and several stomach disorders. Not as efficiently utilized as breast milk. Some babies have difficulty in utilizing certain nutrients of the bottled milk. Bottle feed babies also have the risk of developing an allergy to a particular formula. When a baby develops an allergy to formula, he or she may have symptoms that include irritability, crying after feeding, nausea, vomiting, diarrhea, a skin rash, and these are very expensive. Disorders during embryonic development. Rubella. Rubella, commonly known as German measles, as a disease caused by rubella virus. Rubella virus during pregnancy can be serious. A mother is infected with the first 20 weeks of pregnancy. The child may be born with congenital rubella syndrome. The syndrome follows intrauterine infection by rubella virus and comprises cardiac, cerebral, ophthalmic, and auditory defects. It may also cause prematurity, low birth weight, anemia, and hepatitis. Abnormal neural tube. Neural tube defects are serious birth defects with symptoms that range from mild to severe impairment. They are caused by incomplete development of the brain, spinal cord, and are their protective coverings. This occurs when the fetus spine fails to close properly during the early stages of pregnancy. Thyroid gland. In newborn infants with congenital hypothyroidism frequently have hyperbilirubinemia and delayed skeletal maturation, reflecting immaturity of liver and bone, respectively, and they are at risk permanent mental retardation. If thyroid hormone therapy is delayed or inadequate, their size at birth, however, is normal. Limb abnormalities. The upper and lower limbs have a large number of different genetic and environment derived abnormalities, some of which can be surgically repaired. Among them, partial or complete absence of limbs or digits are common. Genetic abnormalities related to spontaneous abortions. Approximately 50% of first trimester miscarriages are due to chromosome abnormality in the fetus. An extra chromosome or missing chromosome can cause miscarriage, usually in the first or second trimester of pregnancy, or can lead to child with learning difficulties or mental retardation and birth defects. The chromosome abnormalities involving a missing or extra chromosome are not inherited or caused by an exposure during pregnancy. Instead, they result from a chance mistake in cell division soon after time of conception. This error is random, even that can occur in anyone pregnancy. An inherited problem with chromosome can also cause miscarriage. A parent can have rearrangement, translocation of his her chromosome in which the chromosomes are structured differently. Another genetic cause of miscarriage is change in single gene or several genes on the chromosome. This can cause specific genetic diseases or birth defects. Fetal surgery and detecting fetal development problems. Recent development and improvements in parental diagnosis methods, in particular ultrasonography, have made detection of fetal abnormalities possible. Most defects are best treated after birth. Only a few disorders are potentially treated surgically inside the mother body. Prerequisites for fetal surgeries include selection of those fetuses who might benefit from intrauterine treatment, counseling of the family concerned, and highly experienced multidisciplinary team including prenatal obstetrician and postnatal development. Allometric growth, the term means differential growth and refers to developmental patterns of growth which are not uniform, that is, not all parts of the organism develop at the same rate. An example of allometry is An example of allometry as leg length, the sequence of the changes is consistent among all people. The rate, however, aging. Usually, aging refers to the physiologic changes that occurs in all individuals due to age. The sequence of the changes is consistent among all people. The rate, however, can differ greatly. Both genetics and environmental affect age-related changes. Siblings with the same genetic background can have marked differences and longevity due to environmental factors or secondary aging. The causes of secondary aging may be preventable. Process of aging. Aging is progressive physiological process that is characterized by degeneration of organs, systems, and tissues with consequent loss of functional reserve of these systems. Loss of these functional reserves impairs an individual's ability to cope with physiological challenges such as anesthesia and surgery. Individuals of the same chronological age may differ significantly in the rate and severity of functional decline. Persons who maintain greater than average functional capacity are considered physiologically young, and those whose function declines at an earlier age appears to be physiologically old. Signs and symptoms of aging. There are many different signs and symptoms of aging. Most of these develop gradually and are very diverse, but it should be remembered that it is not possible to diagnose aging based on isolated signs and symptoms alone. Different people possess widely varying degrees of these signs and symptoms. Some of these include an overall decrease in energy and vigor, changes in sleeping patterns, decreased memory, skin and hair changes such as wrinkles, brown spots on the skin, loss of skin elasticity, and hair loss. A loss or decrease in vision and hearing, sexual dysfunction, urinary problems such as incontinence, dribbling, and changes in frequency of urination, changes in menstrual cycle, abnormal obesity and inability to lose weight. Secondary aging processes result from disease and poor health practices. For example, no exercise, smoking, excess fat, and other forms of self-damage, and are often preventable whether all cells change as they age, cells become larger. Their capacity to divide and reproduce tend to decrease. Normal cells have built-in mechanism to repair minor damage, but the ability to repair declines in aging cells. DNA is damaged through the aging process and changes occur in cellular membranes, in enzymes, and transport of ions and nutrients. In the nucleus, chromosome undergo such changes as clumping, shrinkage, and fragmentation. Other changes occur in such organelles as the mitochondria and lysosomes where numbers are reduced, causing cells to function less efficiently. This effect also ties in with a decrease in hormonal secretions. A decrease in metabolism has several effects. Toleration of cold and less. Toleration of cold is less. A tendency to gain weight increases. There is decreased efficiency in body use of glucose. Collagen and elastin decrease in connective tissue formation. Lipid and fat content of tissue change. The total amount of water in the body gradually decreases. System level changes in aging. The gastrointestinal tract or digestive tract in general maintains its functional level well into old age. There are three areas that are the exceptions: altered taste, decreased stomach acid, and decreased blood flow to the liver. In the mouth, there is decrease in saliva, which affects chewing and swallowing. Also, altered taste makes food less palatable. Osteopenia and osteoporosis Both deal with bone loss. The difference is the degree of loss between the ages of 30 and 40. Men and women start to lose bone mass and enter a condition called osteopenia, a decreased or lower mineral content of the bone. Osteoporosis then has greater degree of osteopenia and involves both a decrease in bone minerals and a decrease in bone matrix. Usual aging is accompanied by a lower production of neurotransmitters, but only when the drop approaches 50%. While dementia ensures about 15% of the elderly have severe dementia. The heart of an elderly person could be smaller than the normal due to malnutrition, larger than normal due to severe high blood pressure, or unchanged in size. Usual aging causes the left ventricular wall to thicken in the diameter and length of the aorta. A major artery leaving the heart to increase, fat will accumulate in the heart muscle as a response and increase in total body fat. Infections occur more frequently in elderly and are more often severe to low degree working of immune system. Key points about developmental biology, aging, etc. An organism changes from a fertilized egg into an adult. The progressive changes which are undergone before an organism acquires its adult-like form constitute the embryonic development. Early divisions of the zygote are called cleavages. The extra embryonic membranes that form during the first 2 to 3 weeks of development include amnion, yolk sac, allantois, and chorion. During week 3, the two-layered embryonic disc transforms into a three-layered embryo in which the primary germ layers, ectoderm, mesoderm, and endoderm present. This process called gastrulation involves cellular rearrangements and migrations. The formation of organs and systems during embryonic development is called organogenesis. The first major event in organogenesis is neurulation, the differentiation of ectoderm that produces the brain and spinal cord. Every gene is present in every cell of developing embryo. The nucleus determines the characteristics of individual while the cytoplasm selectively turns on some genes and switches off others. A normal pregnancy lasts 9 months. Each 3-month period of pregnancy is called a trimester. During each trimester, the fetus grows and develops. Implantation takes place in the fourth week of pregnancy. A pregnancy of two or more fetuses is called multiple pregnancy. A multiple fetuses can be the same, identical, or different. Fraternal, identical twins or triplets come from single egg that has been fertilized by one sperm.